Hello, everyone, and welcome to today's webinar, Human Immune System Multi-Omic Studies and the Development of High-Throughput Compatible Activation-Induced Marker Assays. I am Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by BioLegend. BioLegend creates world-class cutting-edge antibodies and reagents across a diverse set of applications for immunology research. With over 30,000 trusted reagents and 100,000 citations, BioLegend is making an impact in labs across the world. Learn more at biolegend.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can utilize this Q&A box and we will assist you directly. I'd like to now welcome our speakers, Blessing Musgrove, Research Associate at the Allen Institute for Immunology, and Tyanna Stuckey, Research Associate at the Allen Institute for Immunology. Tyanna, you may now begin your portion of the presentation.
Hi everyone, my name is Tyanna Stuckey, and I'm a researcher at the Allen Institute for Immunology, and I'd like to thank you for joining us today. Today I'm going to talk about the human immune system multi-omic studies that we've developed here at the Institute and how we're transitioning from doing those high parameter studies to the development of high throughput compatible activation-induced markers using lower parameter instruments and assays. Today I'll start by introducing the Allen Institute for Immunology and what's been the first phase of data acquisition using high-dimensional multi-omics. I'll walk us through some of the key reagents that we're using and some of the places in our pipeline that we're automating the steps to remove operator bias. We'll also talk about some of the panels that we've developed for immunophenotyping on spectral flow and how we've incorporated batch control to ensure the quality of our data. I'll also briefly introduce a preprint paper that we're working on, which is a method that we've developed for cryopreserving whole blood that's compatible with single-cell RNA-seq and the automation of that process, as well as introduce our open access data sets and analysis tools. We'll then transition into phase 2, where my colleague Blessing is going to talk about the development of an activation-induced marker assay using image cytometry and some of the ways we've integrated automation into that process that we hope will be useful in qualifying patient samples for hypothesis-driven studies. Here at the Allen Institute for Immunology, we have this goal of advancing and understanding the immune system, and we're doing this by employing a full spectrum of omics tools to understand molecular pathways and mechanisms that are responsible for the development and progression of disease. And we're doing this with a side set of core values, and those are big science, where we're exploring complex problems in biology at scale; team science, where we believe the teams that we have here are stronger than an individual; open science, where we're radically open with the data that we're collecting and the protocols that we're developing, and we're sharing that, and we do that to build trust and credibility within the science community; and impact, where we're working to accelerate foundational research that's going to lead to a deeper understanding of human health and disease. And our goal is to obtain and publish robust and reliable data that gets us closer to understanding what the immune system looks like in healthy and diseased patients. So it's really important to us that we not only collect robust and reproducible data, but that we do so in a way that's accessible to the science communities that need it the most, and in a way that's translational to the patient. So the Allen Institute for Immunology is interested in researching a broad set of immune spaces, and we're doing this by collaborating with several universities and research institutions across the country. And this has given us access to cohorts that we may not have been able to access under other circumstances. And this also includes some of the healthy cohorts that we're working with currently, and that those patients span a broad range in age. So some of the basic premise of the immune system that we're kind of looking to develop protocols and research into is that the immune system operates in a balance, and sometimes you get hypo states and hyperimmune states. And so what we're doing is looking at surface proteins and biomarkers in that way by flow. We're also looking at secreted proteins by like MSD, proteomics, and Olink data. And then we're also able to look at RNA sequences. So with access to these cohorts, we have the ability to put together up to 7 streams of data that can be used to create a personal immune system signature from a single patient. And these assays that we've developed have been developed to run at scale and are robust enough to run longitudinally. And so I'll focus mostly on flow and some of the proteomics data that we'll talk about in phase 2 with Blessing. And that's where our expertise lies. But if there are any questions about the other assays that we've developed and optimized for our longitudinal studies, I'd be happy to take your questions back to our experts in those spaces. So to kind of walk us through some of the key points in this protocol that we've developed and we've published in STAR protocols. To start, cells are thawed and counted using fluorescence-based cell counting on the Celica MX, and that cell concentration is normalized, and then we plate those cells in a master plate. And so then, we have sample 2 split between flow and RNA sequencing. And some of the important parts of that is that we are doing that in a 23 samples plus 1 batch control. And so those batch control samples were sourced from BioIBT, and so we got 600 vials of cryopreserved PBMCs that were all collected at the same time and from a single donor. And each one of these batches, this batch control is run, and it's a patient that we've or a sample that we've analyzed and have a known sort of signature for. And so we run this sample every time we run a batch of this pipeline to kind of control for process errors. Some of the other things are that we're limiting batch effect by using serum-free media and commercially sourced staining medias and fixed buffers. We specifically are using cell stain buffer and BioLegend fluorofix, both from BioLegend, to control for batch effects for additives that you may add to like PBS or something like that. All of our antibodies are purchased from a single lot, and we purchase those in bulk as far out in expiration date as possible, so that we're not changing lots between different batches for our flow antibodies. And then another important key point is that we're integrating some small scale assisted automation using the Integra and changing up some of the traditional plate consumables. So we're using PCR plates while we're staining to get a tighter pellet. So we're optimizing for cell loss and operator bias doing those things. And so just to again highlight some of the technical components that are key for this pipeline is that all of the samples are barcoded. All of the tubes that we have are barcoded and are tracked in our LIMS system. And then again, those samples are counted on the Celica MX, and that data goes into our LIMS system, where it then does, it can calculate, added media to those cells, so we can normalize those cells to around 10 million per mil. And then we're also, again, have incorporated the Integra to our workflow to remove operator bias. And this also allows us to train new scientists coming into the Allen Institute to be able to work on these big pipeline assays, and that doesn't change our data between, who did it. If I did it yesterday and somebody else does it the next day, the data is going to come out and look the same. And so again, those samples then can be split between the 10x protocols that we have optimized, and then we'll run those. We have developed 4 25 color panels for immunophenotyping that are run on the SciTech Aurora. So again, sort of to highlight the Celica MX, which has been a really important tool in developing these assays and developing consistent data through out the longitudinality of this study. So what we've done is we took some data from 42 batches, through 42 batches, and each of those batches contained the batch control. And what we did is we took that, the average of the viable cell count and the average of the viability for that, and that data stays extremely consistent. And then just to point out that it's been consistent over the last 4 years that we've run this pipeline, and we run, approximately 42 batches a year. So that's quite a few in the last 4 years. But to just pull out some data from the last pipeline that was run for us, we ran batch 232 on September 24th of 2024 this year, and that viable cell count stayed within that range that we had from that batch control. And so it's really important to us that not only do we produce high quality data, but that it's consistent through this time, because if you are running an assay that's inconsistent, the results of those can ultimately be really questionable. And so looking through at the flow cytometry data, those 4 panels that were developed, they all have cross-panel markers to identify key cell subsets. And so we're looking at T cells, B cells, NK cells, monocytes, and dendritic cells through that. And this image here is pulled from one of the panels that's really able to identify all of these populations really well. But looking at 10 batches over a few months that this was run, and looking at that batch control, those populations stay really similar. And so that we kind of know that batch control is really working for us. And then the quality of that data is also replicated in the RNA-seq data that we're looking at here. And so what we've got is all of the cell types are able to be pulled out of that data. And then the UMAP you see in the center of this slide is showing 9 batches overlaid, run from May to October, with a less than 0.1 batch variance. And then again, just the far side, this bar graph showing that batch variance is really low, while on the other side you see biological variance between samples, which is what we're looking for. And so moving forward, we have developed this small scale automation, assisted automation, but we have the opportunity to move forward to large scale automation. And so we're doing this on the TCAN, but a lot of this stuff can be done on really any large scale liquid handling robot. So my colleagues have scripted and validated several other modules that can be used in our pipeline and in other cell-based assays. And so for our pipelines now, we have fully automated cell counting and normalization, along with the cell plating. And then we have also the ability, to automate cell stim protocols, antibody staining, and other assays such as qualitative or quantitative proteomics for MSD and 10x flex pre-gem generation. So a really good example of this where this large scale automation comes in handy is when we begin to do more neutrophil depletion for downstream 10x protocols. And so a lot of the samples that we get that we process by RNA-seq are neutrophil and debris contaminated. And this is especially true of cryopreserved whole blood. And so when we're working with a small number of samples and sorting by FACS, which is a great option for when you're working with a few samples, it doesn't take that long. It takes us around 7 minutes to clean up a sample. That's great if you're doing a couple samples. But when we need to increase the batch size of that we're running, when we're looking at larger cohorts and building out these assays to be higher throughput, sorting samples by hand is not feasible. And so if it takes 7 minutes to do one sample, the theoretical time it would take to do 96 samples is around 12 hours. And while those cells are still alive, that's just not possible for those to all make it through the process in one day. So my colleagues who are part of my automation team have developed and integrated on deck with our liquid handling robot a magnetic bead isolation that can be done on 96 well plates. So instead of 12 hours, you can do 96 samples at the same time and process that in 30 minutes. And so what's really cool about this is what we've done is you can isolate isolate immune cells from whole blood that is quality cells to go through the 10x flex protocol. And so down here on the bottom, we're looking at the amount of genes per cell. And so this first column, the gray plot here, is what it looks like when we haven't isolated that, but isolated the lymphocytes from that. But when we are able to isolate the lymphocytes, either through the magnetic beads or through FACS, those samples look very similar. We get the same, similar number of genes per cell. And then that's all really compared back to called PBMCs or PBMCs that are isolated using FACS. And so all of that data looks really good. And so what we've done is now we've sort of put out a paper that's being reviewed currently. It's called Cryoscape, and it's a novel whole blood collection and isolation protocol that is compatible with RNA sequencing. And if you're interested in checking that out, there is a link here, a QR code that you can scan that will take you to that preprint. So up to, you can collect this blood. You can collect 2 mils of blood, put it into 2 mils of cryopreservation media, throw it on dry ice, and that can be delivered to us, where we can then thaw the samples and then kind of walk through this protocol. You can thaw the samples and plate them, and then everything else is done on a liquid handling robot. So we'll put it through 2 rounds of magnetic bead enrichment. Then it'll go through high throughput fixation and hybridization. We'll take that then to cell counting to make sure we have good enough, good number of cells, and then through pooling, and then it can go through droplet formation and library prep, and then we'll have data to analyze. So some key points here is that we're using MojoSort magnetic bead depletion, and that's integrated on deck, and then that we're able to cell count those cells in a high throughput way. And the technical coordinates is, very high. It's very comparable each time we've done it. And so after all the 3 years that we've collected this data, we have a lot of big data sets, and we're starting to look through this and publish on this data. But the other thing that we've done is we've put this in a place where everyone can look through these data sets, and also do some of their own research. And so the first set of data that we have released to the public is our Immune Health Atlas, which is looking at healthy patients through age and looking at different cell types between young patients and older patients. So that's the first set of data. We've got a couple more data drops basically coming. And so if you're interested in looking at through those, all of those data apps and data sets and visualizations have been created by researchers at the Allen Institute, and that all goes into a place called the Human Immune System Explorer. And so if you scan the QR code down at the bottom here, you can also take a look at the data that we've produced here. So to follow up on the data that we've collected and the signatures and cell subsets that we've discovered, the next phase here is to mine the data. And so we're looking through this, finding subsets and things that look interesting to us, but we need to find a way to do that in a way that is less expensive and more accessible. And so sort of to move forward effectively, we need to increase the batch size and lower the barrier to access. And we're doing this a little bit by working with the Celica PLX, where we've developed smaller, more targeted panels, or we can develop these smaller, more targeted panels, looking at core subsets of cells, and develop agonist screens that can be analyzed using this instrument. And the overall, the big thing that comes out of this is the reduced cost per sample, where running these high parameter flow assays and sequencing costs way more than that. But we're taking this from $120 per sample to around $10 per sample, which is much more accessible to places that don't have the kind of funding that the Allen Institute has. And with that, I will hand it off to my colleague, Blessing.
Hi, my name is Blessing Musgrove, and I am a research associate at the Allen Institute for Immunology. In this section, I'm going to focus on Phase 2. As my colleague Tyanna mentioned, Phase 2 is where we are mining the data, aiming to target lower dimension and increased throughput multi-omics for hypothesis-driven studies. Therefore, I'm going to discuss how we developed an activation-induced marker assay using image cytometry. To help us develop our assay, we used the Celica PLX. The Celica PLX is made by Revvity. It is a high-throughput image cytometer that is capable of bright field and fluorescent-based cytometric analysis. The PLX is a multiplex system. It has a blue, green, orange, red, and far red fluorescent channel, as well as a bright field channel to count and identify cells, meaning that it is capable of direct cell counting, determining cell viability, detecting apoptotic cell health, fluorescent protein expression, and immune cell phenotyping. During our assay development, we compared the PLX to a traditional cytometer, and in this case, we used the Beckman-Coulter Cytoflex. Unlike a traditional cytometer, the PLX does not require instrumental setup, cleanup, and shutdown, saving time while running an experiment. Also, the PLX uses just an aliquot of sample, meaning that you can take an aliquot of your sample, run it on the PLX for evaluation, while using the rest of the sample for downstream processing. Before developing our assay, previous experiments were performed on the PLX to determine if the PLX can analyze and detect immune cell core subsets, and how the results from the PLX compare to a flow cytometer. Here is a paper that was published in collaboration with Revvity that was comparing the Celica PLX image cytometer to the spectral cytic Aurora, and it was using PBMC samples from 24 patients. This paper was looking at population percentages of important immune cell core subsets, CD3 positive, CD14 positive, CD56 positive, and CD19 positive cells detected by each instrument. Results from the PLX are in red, and results from the flow cytometer are in blue. Results show that the PLX is capable of detecting core immune cell populations, and that is comparable to a flow cytometer with a coefficient of determination of 0.98. After knowing that the PLX is capable of detecting core immune cell subsets, Tyanna and I wanted to focus our assay on T cells, specifically on T cell activation with PHA. We wanted to compare activation of healthy patient T cells to that of diseased patient T cells, and we decided that rheumatoid arthritis would be the disease that we focused on. Here at the Allen Institute for Immunology, we study many autoimmune diseases, and rheumatoid arthritis is one of them. There is an unmet need in understanding the molecular drivers of autoimmunity, especially when trying to understand preclinical and early RA. We are mainly trying to understand, is T cell dysregulation or reduced response to TCR cross-linking a trigger point? And can we build a robust automation compatible AIM assay to investigate this? To help us understand and answer those questions, using the Celica PLX, we optimized a panel to monitor activation in T cells. The panel we optimized was a 4-color panel that consists of T cell activation-induced surface markers that would help quantify the response between healthy and rheumatoid arthritis patient samples. So, why is this important? It is important because the modular activation panel that we developed can be used to quickly monitor cell subsets during treatment of a disease. The methods that we use are compatible with automation, which helps minimize user error and ensures reliable and consistent results. And the assays that we developed are completely transferable between the image cytometer and a traditional flow cytometer. Furthermore, at the Allen Institute for Immunology, we are committed to open and accessible science. The Celica PLX helps make our science easily accessible and more open by being user-friendly and not requiring any startup or shutdown procedures. It doesn't have any fluidics. It has fixed lasers and outputs flow-like data with a templated data analysis, making it easier for anybody to use and analyze their data. So, what did we do and how did we do it? We developed a multiplex detection method to monitor T cell activation using a 4-color panel consisting of the surface markers CD3, CD69, CD25, and CD279. We used CD3 because it's a T cell lineage marker. CD69 and CD25 are activation markers, where CD69 is our early activation marker and CD25 is our late activation marker. CD279 is PD1, the checkpoint and exhaustion molecule, which is disease-specific and would hopefully show a difference between healthy and RA status patients. To stimulate the cells, we used PHAP. We obtained isolated cryopreserved PBMCs, cultured the cells with titrated PHAP for 24 hours. After 24 hours of incubation, we removed the supernatants for a cytokine assay, and then we measured the cytokine concentration via MSD technology, which I will go into more detail later on about MSD. After the supernatants were removed, we stained the cells just like we would stain cells for traditional flow cytometry. We used best practices in flow, and as well as the same reagents such as BioLegend's antibodies, FCX TrueStain, and we also fixed the cells using BioLegend's FluoroFix. Also, we integrated automation for liquid handling to maintain consistent results. Once the cells were ready, we took an aliquot of the cells and loaded them onto the PLX, and we optimized the exposure time for each fluorescent channel. And because the PLX also has a microscope component, we made sure that our cells were in focus, and then we ran the cells. Once the cells were done being ran on the PLX, we loaded the remaining cells left in the plate onto our traditional cytometer, the Beckman-Coulter Cytoflex, to be able to compare the results. Below, we have added the fluors that we have tested and are using, which we have sourced our antibodies from BioLegend. However, if you have any questions, Brevity likely has suggestions that you can use. We have chosen these conjugations in our panel because they are cataloged and ready to purchase. They are less expensive than custom conjugations or tandem dyes, and that was and will be a factor for how we develop assays for this instrument in the future. When a sample is finished running, this is an example of images the PLX takes from each channel. These are PBMC samples from a healthy donor where we are comparing an unstimulated sample with a stimulated sample that has been stimulated with PHAP for 24 hours. The PLX shows you bright field, which is used for counting the cells. We have our CD3 where we use KB520. For CD69, we used BV421. For CD25, we used APC. And for CD279, we used PE. And again, we sourced all of these antibodies from BioLegend. Looking at bright field, you can see the difference between the unstimulated and the stimulated sample. The stimulated sample has clump cells, while the unstimulated sample doesn't. And as you can see with the unstimulated sample, when looking at our two activation markers, CD69 and CD25, we don't see any cells expressing those markers, while in the stimulated sample, we see an increase in expression, and we also see an increase in expression of PD-1. This is cool because it allows you to be able to see which cells in your samples are expressing certain surface markers. And here is a representation of the data analyzed from both instruments. Because the PLX outputs flow like data, we were able to analyze the data in FCS Express, which allows us to compare the data from the PLX with a traditional cytometer. During our experiment, we did a full titration of PHAP using healthy donor samples. Here we are showing results from one of those healthy donors. We compared an unstimulated sample to a stimulated sample. We gated on CD3 because we wanted to only be looking at T cells, and that is what the histograms are showing. After gating on CD3, we gated on the other three surface markers, and we have shown those as density plots. Looking at our activation markers, as well as our checkpoint and exhaustion molecule, we see that both instruments yielded comparable results. Orthogonally, cytokine release was measured using the VPLEX pro-inflammatory panel 1, which measures the concentrations of the cytokines listed. The assays in this kit are sandwich immunoassays where a capture antibody is bound to the plate, the analyte is bound to the capture antibody, and then the sulfo tag conjugated detection antibody is bound to the analyte. Voltage is applied to the plate, and the light that is emitted by the detection antibody is measured. Using a standard curve of known concentrations, we can measure the amount of cytokine released by the cells that went into culture. In this assay, we employ a similar set of standards to control for technical and biological variation. The standard curve controls and samples are run in duplicate to control for technical error, and automated plate washing and liquid handling are employed to ensure accurate results. Although we are looking at a full pro-inflammatory panel, because we wanted to specifically focus on T cells in our PBMC samples, the cytokines we were most interested in were IL-2, which is a glycosylated protein produced by T cells, and it supports T cell growth, IL-4, which is produced by Th2 cells, and interferon gamma, which is produced by Th1 cells and is an activator of macrophages. However, after analyzing the data, we decided to move forward with only IL-2 and interferon gamma because IL-4 stimulates B cell growth and suppresses Th1 cell function, which meant it wouldn't be a helpful cytokine for us to focus on. And because the PLX outputs flow like data, we were able to obtain the median fluorescent intensity, or MFI, for each marker. Using PBMCs from healthy donors, we titrated PHAP, gated on CD3 to make sure we were getting all our T cells, and graphed the MFIs for both our activation markers so that we could obtain EC50. EC50 is the half maximal effective concentration. We were hoping that from the EC50 value we got, we could use that value moving forward to test for activation between our healthy and our RA samples. Comparing the EC50s for both the PLX and the Cytoflex, we saw that the EC50s for both CD69 and CD25 after 24 hours were comparable. On both instruments, we saw that there was concentration-dependent increases in the activation markers, meaning that as the concentration of PHAP increased, the expression of the surface markers increased until the concentration of PHAP became toxic to the cells. We also graphed the MSC data that we got using the supernatives from the cells. We were able to obtain cytokine concentrations of interferon gamma and IL-2. We were able to also get EC50 values for both cytokines, and like the activation markers, we saw concentration-dependent increases, meaning the higher the concentration of PHAP, the more cytokines were secreted. After determining that we could obtain EC50s from the PLX that were comparable to the Stataflex and also showed the same trends as the results from the MSD assay, we decided it was time for us to incorporate our rheumatoid arthritis patient samples. We stimulated cells from 12 healthy and 12 RA patient samples for 24 hours. After 24 hours, we took the supernatants for our MSD assay, and then we stained the cells using best practices in flow cytometry. And once the cells were stained, we ran the cells on the PLX. Here, we are comparing our unstimulated samples to our stimulated samples. When comparing the reactivity of the healthy samples to the RA samples, looking at the two activation markers, CD69 and CD25, the healthy samples were more reactive than the RA samples. We also noticed that the healthy samples had a little higher expression of CD279 when being stimulated, which was interesting because we weren't sure if we could see any difference between our healthy and RA patients, and because we know that CD279 is disease specific. Looking at our interferon gamma and our IL-2 cytokine concentration, like the surface markers, the healthy samples had higher cytokine concentrations compared to the RA samples, showing that there was a reduced T cell response to TCR cross-linking in the RA samples.
Thank you, Blessing. So to summarize, through our core values of big, open, and team science with impact, in phase 1, we developed protocols and assays that are robust and consistent to generate data that we're publishing openly and sharing with the scientific community. We've integrated automation to continue with our studies at scale, and researchers here at the Institute have developed and generated open access tools and data sets to comb through this data. In phase 2, we took a stab at more targeted assays using lower dimensional assays developed on the PLX and determined that we can still get really good looking data that's comparable to its counterpart in flow, and while still doing these assays in a high throughput way and at low cost. And with that, I'd like to take a moment to thank our collaborators and members of our cohorts who, without their samples, none of this data that we've generated could be possible. I want to thank our team at the Allen Institute for Immunology. Thanks to those who developed these assays, those who are processing these samples with care, and to those who have supported our work. I want to thank our communications team who helped us promote this event and created nearly all of the QR codes you saw throughout the presentation. And I'd like to thank our friends at Revity for helping us incorporate the Celica MX and the Celica PLX into our workflows. And thanks to our friends at BioLegend for supporting the development of these assays. And thanks for inviting us to speak today. Leave you with a thank you slide and a QR code that will take you to the Allen Institute for Immunology's website, where you can find all of the papers and open access data sets and tools that I've mentioned today.
Thank you for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question located on the lower left corner of your screen. We'll answer as many of your questions as we have time for. All right, let's go ahead and get started. Our first question asks, what is one of the main challenges that you had when developing the panel for this platform?
Yeah, so I'll take a stab at that. So unlike traditional flow cytometry, we have exposure times to deal with rather than gain settings. So that was a little bit of a challenge. I think both of Blessing and I come from flow backgrounds, and we haven't done a lot of like image cytometry or microscopy. So kind of taking that stab, going right into it and saying, okay, this is how we would do it on flow, and kind of changing that up. So some of the things just to remember with this platform is that you've got exposure times to deal with. So starting out sort of relatively high and then titrating that exposure time down is sort of the way that we developed those panels, and kind of got really surprisingly good results from it. What we were expecting that looks similar to flow.
Great. right. Moving on to our next question here. This question asks, how do you deal with cell counting on clumped cells on the PLX?
Sure, I'll take a stab at this one. So the PLX has a cell counting algorithm that is able to detect the edge of a cell, which allows it to count the majority of the cells in a clump. But also the fluorescence algorithm is able to normalize the fluorescence intensity of the cells based off of cell size, which helps keep our MFI data from being skewed, and for it to be consistent and reliable.
Wonderful, thank you. All right, please go ahead and keep submitting those questions so that we can ask them for our speakers who've joined us today during this Q&A session. We have one more here in the inbox that says, you mentioned that the PLX is cheaper than traditional flow cytometer. How is it cheaper?
Yeah, so the PLX doesn't have any fluidics, so you don't have to worry about contaminating your cells and potentially having to run your samples again because of contamination. It also doesn't require any single stain controls, which allows you to save on sample. And because it only uses a 4-color panel, you're able to save money on those antibodies. And then also, because it doesn't have any shutdown or startup procedures, you save time, which then in the long run saves cost on labor.
Perfect. All right, we have another question here that was submitted. It says, in terms of accessibility, how do you think the PLX compares to other similar instruments or other cell counters?
Yeah, so this instrument, unlike other like traditional flow cytometers, doesn't require any fluidics or startup protocol. And so it kind of saves on training techs to use the instrument. I mean, Blessing and I sort of run the not a core, but run the flow cytometers at the institute, and they are complicated instruments. And this instrument, the PLX, is a lot more user friendly. The other thing that I think, just like accessibility in general, is that it doesn't have it's fixed laser alignment. So again, not having to train a user sort of in that like background fluidics and optics technology, but it's small and it can be taken places that a flow cytometer couldn't be taken. And so I wrote down a couple of things because while I was listening to this presentation, I thought these were a couple cool things. I went to a university that had one instrument, a Biorad S3E, that was to support all of the research that was happening at the university, and that meant it was really hard to get onto those instruments. And so this would have been a really nice thing to have in a low-funded university. Also, a couple of the things that this instrument is really good at is pulling out populations, and I think something that it could be really good to use for just example is to like monitor CD4, CD8 T cell populations in HIV patients, and being able to do that all over the world or at a, outside of a, in a parking lot kind of thing, get samples, process them really quickly in a pop-up facility. Whereas the samples that we're analyzing here are processed in facilities that have the capability to do the like phi calling of samples. So it ties in really well with the other work that we're doing here at the Institute and just like trying to make science accessible to everyone.
Sorry, I was muted. Thank you so much for that answer. That was very great. If anyone has any additional questions, now is the time to go ahead and submit those. Before we wrap up here, I wanted to just ask either of you if you have any closing statements regarding the presentation, any last pieces of information that maybe you didn't get to share.
Just, that we're really appreciative to have this opportunity and to share the science that we're doing. I think the more we get the stuff that we do out in platforms that, are being invited to, I think it's a great opportunity to share what we do here.
Okay, wonderful. All right, I don't see any more questions that popped up into the inbox, so we're going to go ahead and wrap here. I want to thank you both for your time today and your important research. We would also like to thank LabRoots and our sponsor BioLegend for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. So please continue submitting your questions as they will be responded to. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you and take care, everyone. And goodbye.